생물고고학 도구로서의 고대DNA라는 주제로 발표를 해주시겠습니다. 아주 흥미롭고 유익한 시간이 될 것 같은데요. 로드리고 바르케라 연구원님을 박수로 모시겠습니다.
Good afternoon, everyone. Thank you so much for having me here today. This is a bit of a challenging talk, so I want to present what ancient DNA is, how you can use it for better archaeological and bioarchaeological, specifically bioarchaeological research, and the challenges that this would bring when you try to incorporate ancient DNA into your studies. Because it's not an easy task just to say, okay, we're going to do ancient DNA. It has to do with a very careful assessment on what to do, because this is a semi-destructive method. We cannot only analyze it like traditional archaeology or traditional osteology, in which we only look at the object and then we say something about it. We have to sample the object, we have to drill it, we have to cut it. So we need to assess exactly the needs, the questions of all the stakeholders before we make any decision on how to proceed with the sample. This story is very interesting though, because it's, as any good, very cinematic story, it starts very slowly, and it's just like the development of genetics, when we can see that, of course, we have the first insights came from Mendel and his piece, and those were the foundations for genetics and even modern genetics and genomics. But it took quite some time between these started and the first molecular studies. So when we were dealing with the structure of the double helix of DNA, we already knew that this molecule was important. We didn't know back then that the genes were living there, but we had an idea that maybe those entities were living in this double helix structure, which actually is very important for our field, because only this configuration, the double helix, is the one that can be present in all the elements that we analyze after very long time periods. So it's not just by chance that we can analyze ancient DNA. It has to do with this structure that in 1953 was first reported and granted the first Nobel Prize for this, let's say, for this field, for genetics. And we won't see much other advancements until the end of the 20th century, with the development of the PCR, and PCR became widely known during the COVID pandemic, because that's one of the diagnostic methods that we use for COVID and other viral diseases. And we strongly rely to this day on PCR to prepare our libraries and everything that will develop into the field of ancient DNA. And if we take a look at the last decade of the 20th century and the first years of this century, we had a big landmark, which is the report of the first draft of the human genome. And that was a strategy that costed a lot of money, it costed a lot of effort, but in the end it gave us one of the most amazing stories ever, which is the biography of our species. And for the first time we could look at the genes and ask questions, evolutive or evolutionary questions, and also adaptation. We could ask history to these genes. So it was quite a landmark to get to this point. Now, this field started a little bit before we could complete the first draft of the human genome. So in 1990, we first started drafting our genome, and some years before, the first molecules, the first ancient molecules, were successfully recovered from both human remains, mummified human remains, and also from animals that were sitting in museums. And the characteristics that we learned that they had were damage, mainly the deamination, which is when we lose part of the structure of the base pairs that conform the DNA. And also, this deamination creates sites that are more prone to hydrolysis, so DNA can break, and then we don't have a full-length molecule where it starts breaking down. On the other hand, we also have depurination, and then we can map this as damage. So this is a way in which we could identify that these are actually ancient DNA reads. But the technology for assessing this damage as part of the molecule itself, and in a way we can authenticate that this is ancient DNA coming from an ancient sample and it's not contamination, was not available when we first started in this field. Back then, we only had Sanger sequencing, which is what was used to map the first draft of the human genome, but it's not cost efficient and it's not time efficient. So up until 2007, around 2007, Illumina asked what can we do to start doing ancient DNA in a more straightforward way, like not depending on the Sanger sequencing, which is very slow, but being able to sequence massive amounts of DNA in a short time of period. So then we had what we call next generation sequencing, and from that point onwards we have been able to sequence many more sequences per day, but also the costs dropped significantly. So we went from around $100 million by the time that we reported the first draft of the human genome. That's the cost of one genome. And right now we're about somewhere in between $100 and $1,000 per human genome. And we can now sequence it in 2 days. That the sequencing only takes 2 days, compared to 1 decade before. This, of course, allowed us to get a lot of ancient genomes coming from samples from archaeological contexts. Right now we are around, or more than 20,000 individuals spanning 100,000 years of human history, on more than recovered from more than 2,500 archaeological sites, which is a lot, but still you can see there are many places, there are many regions, full regions missing. We're going to address some of the questions on why are these regions not studied enough, and that has to do not only with the limitations of the field, but also the problems that we're facing when we're trying to incorporate this field into archaeological questions. But we have a beautiful opportunity here, and we have what I like to call a molecular time machine, which is similar to the one that is described in the series Cosmos. If you have seen it, you will understand about this spaceship. If not, do yourselves a favor and watch it. The series is amazing. And with this little time machine, what we could do is travel back in time and first report of ancient DNA came from now classical paper by Higuchi and collaborators describing the ancient DNA sequences of the mitochondrial DNA from this animal here, which is called quagga. And remember, back then we were just dealing with mitochondrial DNA, so a very small fragment of the whole information, genetic information of a full organism. But even then, with this little amount of information, we could tell something very interesting. This animal that looks like a zebra, it's actually related to a zebra. And we could say, okay, we didn't need a test to actually relate this animal to the zebra. We can see from the picture it looks like a zebra. But it's not always the case in science. We will not only think that things that look alike are related. So it was important that we could test this, and it was sort of expected, the result, but also it was a control, because we were expecting to have these results sort of. So when the first team reported ancient DNA coming from this sample, they already knew what to expect. So this was, in a way, positive control of something that we were expecting to happen, and it happened. So this opened officially the field of ancient DNA. And ever since, we can see that terms like ancient DNA, paleogenomics, archaeogenetics, all these terms that are related to the same thing, but they are slightly different in the definition, have been growing steadily since the very beginning of the field. And even before, we already knew that there was something there. We just couldn't name it. Interestingly, when we compare, this is books on the upper graphic, and below we have search terms on Google. So we only have from 2004 onwards, but we can see that ancient DNA as common knowledge is much more frequent as a term in the academic fields. So people in academic fields would use archaeogenetics, paleogenomics, not always ancient DNA, but common people would search on the internet for ancient DNA. We have an issue there already, because how can we communicate efficiently what we found on paleogenomics if the people are searching for ancient DNA? We have to bridge those terms. And a lot of this talk is going to be about bridging things, bridging fields, bridging communities, and also terms. And so, as I was saying, the number of publications has risen steadily. This is just until the end of last decade. We can see the number of publications. This is not cumulative, so every year there are more and more papers coming from the field of ancient DNA. And it's very interesting, and people has a lot of interest in these stories, because it's basically this story of our species, our own story. So of course, one has to always ask questions when a field is born, and when we are taking directions on this field. Do we want to keep going the way it is? Do we want to turn and do something different? Do we want to incorporate other fields? And this put us on a crossroads. And we have to make decisions, and I think that we are at this point right now. Maybe we're a little bit past this point, because we have already started doing research into one specific road, which is an interdisciplinary approach to the study of the past. And of course, one of the first questions that we asked when we finally could prove that this molecular time machine was working was, are these other hominins that we have found ever since like 19th century, these Neanderthals, are we related to them? They look so much like us, but are they actually like us? Are they our ancestors? Are they sister clades of our human species clade? And some of these questions were answered fastly. So the first one was, and again, this is part of the field, of the development of any field, we had mitochondrial DNA, and the first question was to see whether the mitochondrial DNA of Neanderthals would resemble that of humans or not. And the answer was no. Neanderthal mitochondrial DNA is not the same as human us DNA. We cannot find any of this mitochondrial DNA in modern populations. So back then, the answer to the question whether we are related or not was no. However, when we studied the full genome of these other people, we found that we actually carry signatures of these genomes from Neanderthals. Not only that, then we found another species that was only the tip of the pinky finger and one molar, and that was it. That's the entire fossil. And that was a new species. And we could say, okay, this species that we don't know how it looked like, because all we have is this. And as good as we are right now with reconstructing faces of dead people, we cannot reconstruct the full face from this only. But we could tell, okay, there is a percentage of our genome of what we are today that belongs to this other species, the Denisova. Not only that, now we know that there was a fourth species, so humans, Neanderthals, Denisovas, and a fourth ghost population for which we have no fossils whatsoever. But we know that there are traces of this genome in our genome. So we know that other species also contributed to what we call humans today. And we still don't have the fossils, so this is very, very exciting. Another exciting thing that we learned is that, and try to imagine this as lottery. If you play lottery, imagine winning the largest prize ever. You have one fossil, and this fossil is very, very precious because you don't find them all the time. And this girl, this pre-teenager aged girl, was shown to be the first generation daughter of a Neanderthal mother and a Denisovan father. When we color the genome, you can see two colors here. So blue is Denisova, and then in red we have Neanderthal. And if we take a look at the whole genome of this individual, is purple. So basically both genomes in the same proportion. This is a first evidence that not only humans were interbreeding with other species, but also these other species were interbreeding with each other. Sometimes we have, in the case of Neanderthals, we don't have a lot of insights, we don't have any written language, we don't have a lot of material culture that can be ascribed to this species. Sometimes we have them, but it's not that easy. But for some of the other examples that I'm going to refer to in this talk, we do have archaeological records and even historical records, and those are also ways in which we can tell the whole story. This is also another very exciting project, and in this case what they were doing is analyzing individuals that were buried in a cemetery that was signaled with tombstones saying pestilence. And for very long, the academics, specifically historians, have said that in Central Asia there is a place that could have been the origin of the Black Death. But this was, again, subject to interpretation because historical records, and we never know how accurate they are, and pestilence can mean many things. It's not only one disease. Pestilence is one of the most widespread words, and even the name of the bacterium that causes the Black Death, Yersinia pestis, has the pestis in its last name. So it's complicated to say yes, it was only like that, just by the name in the tombstones, but when they could recover ancient DNA from these individuals that were buried in these specific burials with the tombstones, what we could see is that this Yersinia that was recovered from this site in Central Asia is the mother of the Black Death and all the other descendants from this Black Death pandemic event, including the second plague pandemic. So we are seeing the birth of a pandemic, just as we did in 2020 with COVID. So this is just to say, studying the past, of course, has implications for the present and also the future, because now we can understand how these pandemic events develop, not only for viruses, but also for bacteria. There are many, many examples on how to incorporate ancient DNA into archaeology and history. I could spend days talking about many papers dealing with a lot of these examples, but I'm going to focus on some of the ones that I have had the privilege to work in and being involved in the development of them, because then I can answer questions directly from these works. One of them pertained Africans that we discovered in early colonial Mexico, a mass burial that was supposed to be a hospital in what today is Mexico City, in downtown Mexico City. And this burial was supposed to be specific for the indigenous Americans of the region. The name of the hospital was San José de los Naturales. The Naturales, naturals, are the indigenous peoples. So this was a hospital that was devoted by the Spanish crown to only take care of indigenous people. So what were they doing, 3 African individuals there? We will go there. First of all, the archaeologists and the anthropologists already said this could be that these individuals are not indigenous Americans because they look taller and stronger in a way than regular indigenous American skeletal elements. So they already knew only from looking at the bones, they may not be indigenous Americans, but they could not say specifically, okay, they are from a different continent. So what we did first was to assess whether they were or not, in fact, indigenous Americans, and what we could see is that genetically speaking, they belong, these individuals, to groups that are present in what today is Western Africa, Western and South Africa. So they were genetically Africans. Okay. Second thing is, were they born in the Americas or were they born in Africa? Because that already changes the story of what we're seeing here. And with the aid of strontium isotopes, we could answer that question, and we said, okay, they were actually born in Africa and in Western Africa. So the genetics and the isotopes completely match. That's perfect. So these individuals were indeed first generation Africans. From the osteobiography, we could tell that they were mistreated heavily. We can tell that they were shot at. This is what you can see here, this greenish color is because they had a gunshot there, and the gunshot itself was made of copper, and the copper, after the years, stained the bone with this copperish color. We could see in another individual poorly consolidated fractures consistent with them being abused and then not taken care of. And you can see this, of course, created a lot of stress, mechanical stress on this individual. But you can see, because this healed, that this is not the thing that killed him. Also in this case, we can see the fracture in this rib, but this is not the thing that killed this individual. And we can see here muscle insertions, so the places in which the muscles attach the bone. And if you work out a lot, or you're subject to a lot of stress, then you can see that these insertions become very apparent, and you can see here it's super strong, these insertions in which the muscles attach, and this is indicative of a lot of stress. Again, mechanical stress. All of this is consistent with them being enslaved individuals. We also found that they were affected by chronic diseases, chronic infectious diseases. One of them was affected by hepatitis B, and the clade in which the hepatitis B was found was also that of other hepatitis B strains that are common in Africa, again consistent with the whole story. And another individual was affected with relative of syphilis, so this is JOS, a different disease. And we can see again that this individual here clustered together with some strains that are common in Africa. Again, all these would say these individuals were born in Africa, got infected in Africa, were enslaved, transported to the Americas, and they died there. But interestingly enough, again, they didn't die of all the abuse. They died young, between 25, 35 years old, but the abuse was not the cause of death. And this was a mass burial. So we think this was an infectious disease that was not the infectious diseases that we already identified, it could have been a virus that is not DNA based. Now we have, since we're talking already of colonial Mexico City, I'm going to quickly go through this. So we have this story that is very famous of this in the Americas, that is when the conquerors, the Spanish army came to the Americas and they tried to conquer central Mexico. We know that they defeated the so-called Aztec. It's not the proper name is Mexica. And they defeated the Mexica in 1521. If you look, in 1520, there was smallpox epidemic. So we already know that this decimated the inhabitants of central Mexico, and that's why actually Cortés could conquer central Mexico. He was aided by diseases. But there were two other very serious events that we could not tell what were they, and no one could tell up until this moment, because all the symptoms are very difficult to put together in one specific disease, and no one knew what was that. So the Spanish crown, the proto-medic for the Spanish crown didn't know, and the Mexica themselves didn't know. We have one candidate right now, which is Salmonella enterica paratyphi. It's not the same bug that affects the gut. So if you eat something and then you get food poisoning, it's not that. This is enteric fever, so it's a systemic disease and it's much more serious. In a very elegant study, some of my collaborators at the Institute analyzed pre-contact and post-contact individuals from the same place. This is a place that is in southern Mexico that is called Tepozcolula, and they were comparing before and after contact individuals just to see if they would find any pathogen that would point to these Cocorlisli events. And what they found is that the pre-contact individuals were basically clean, they would not have any pathogens, but post-contact individuals had a lot of these reads mapping to Salmonella genome, meaning that Salmonella probably was one of these causes for this disease. But you can say, you cannot tell a story like that and saying that with one point in time and space. You need more evidence. And I agree. We have now a second place that is not close by, it's in a different place. Now we're talking about Mexico City, southern Mexico City. And in there we found again Salmonella enterica, the same species, the same strain, Salmonella enterica paratyphi, in many individuals, now from a different place and from the same time period. So this is a little bit more convincing. And these individuals are, even though they are from the beginning of the colonial period, they still do not exhibit any admixture with European or African sources. This is the genetic makeup of these individuals. The little stars that we can see here are in the background of other Native American populations. Here in green we have Europe, and this is the African diversity, and we can see that they all cluster here, meaning they are all indigenous Americans. And when we compare the strains that were recovered, or the genomes that were recovered from Tepozcolula, so the first site, we can see that they all are basically the same thing. So the new site, San Gregorio Tlapulco in red, clusters together with Tepozcolula, but also clusters together with these two medieval European strains of the same pathogen, which further supports the idea that first this strain originated in Europe from the medieval period already, and then it was transported to the Americas, because that's the only explanation for them being so close together, genetically speaking. But you can say again, they look alike. What if they independently evolved, but they are not related? Okay, fair enough. We have another test, which is to compare the presence and absence of genes. This is not single mutations. This is full genes that are organized in what we call pathogenicity islands. And we have 10 of these pathogenicity islands for Salmonella, and we can see that the pattern of presence and absence, so this is absent, and yellow is present, and we can see that the presence and absence of these full regions, again this is not just one mutation, it's a full region, is exactly the same for the 3 sites that we already analyzed, and at the very top we can see paratype C, so this is the reference strain, so we are completely sure they are all the same thing. What is most? We would expect if such a large epidemic event happened, then the survivors must carry a signature in their genomes, right? Like the survivors are expected to have something that made them survive. And we actually found that. So there is one position in our genome, in the chromosome 6, that is called HLA, which is this molecule. It's a very nice molecule. We'll have different versions of these molecules. If I would genotype all of us right now, I could bet whatever you want that none of us have the same information in those genes. Absolutely no one. Which is why it's very difficult to find a match for bone marrow transplants outside of the family. That's the reason, because this is very diverse, is this system. And what we can see here in this 3D picture, this place is where we put small fragments, in this case, for example, of Salmonella, and we present that to the immune system, so the immune system can start an immune response against whatever, in this case, Salmonella. So we depend on this system to start our immune response. So of course, if we find a variant that is good for protecting us from developing, for example, Salmonella fever, then that variant should go up in time when we go from the event to the future, right? We had the opportunity to work in one place in which we could compare 2 populations that are genetically the same in the present and in the past, which are individuals from the Maya region. Now we're moving to Southeast Mexico, and in there we have a place that is called Chichen Itza, which I'm sure that you have heard of. And these individuals from Chichen Itza and present-day individuals from Tishkakatuyú, they share the same genetic ancestry. They are basically the same. So we can compare things and see what happened after before and after the contact with Europeans. What we can see is that one of these specific variants for these HLA molecules, one of them went from 23% to 46%. So it doubled the frequency through time. And this variant is actually the one that has been reported to be resistant for enteric fever. And again, you can say, okay, but this is just one shift. Maybe it's not that story, maybe it's something else. Well, okay. What if we map the frequency of this variant in the whole world? We can see that in all the places in which the Cocolisli was reported in historical records, we have the highest frequencies of this allele that is resistant to Salmonella. So this adds up to the whole story of not only Salmonella being the agent that caused this Cocolisli, but also it left a mark, a footprint in our genomes, and we still carry that to this day. I have that variant, so I am resistant to Salmonella, not all the time. Sometimes I go to Mexico and I eat some street food and I would get sick because of Salmonella, but I'm never as sick as if someone without this allele would go and do the same. I don't have a lot of time to explain a little bit more from this study from Chichen Itza, but I'm going to jump to the last part. This was a full other story in which we found that these individuals that we were studying in Chichen Itza were actually related. So this is a burial that was a sacrifice, but it was not a sacrifice from a single occasion. They were putting their these individuals. They were all of them kids between 3 and 6 years old. But interestingly enough, and this is one of the things that Dr. Jessica Liu was pointing out before, we cannot call the sex of these individuals confidently just by looking at the bones. But when we have genetics, we can call the sex, and all of them were male, which already tells that there is something going on there. They were selected for. And not only that, we also knew that there were pairs of twins, which would relate to Mayan cosmogony. And the most important thing that I want to go through before finishing my talk is that we consulted with Tishkakal-Tuyuk. We already said that they are related genetically, so we went there, we explained the results, and we consulted with them. What do you think about this story? Are you happy with this story? Do you think that we should add something to this analysis before we publish it? And they were really engaged with our results. They were happy to learn that they were related to the people living there years ago. So on that, I want to finish saying that one of the main challenges that ancient DNA is facing today is backlash, because not all the time we include the communities in the speech in which the speech that we are putting there with our stories. So we want to consult with communities, we want to hear their questions, because they even have very interesting research questions that we couldn't figure, because it's always better to have more brains studying the same thing. And we're trying to incorporate their knowledge, and we're trying to share what we do with them, with some strategies for community engagement, like these coloring books, which we translate into every language of the regions in which we're working. So this is aimed for kids, but also adults will read them, and they will learn, and they can ask questions, and then those questions can be part of our papers. With that, I want to say thank you so much. I would like to pronounce that in Korean, but my Korean is not good. But thank you for your attention, and I am here for you if you have any questions.
네, 로드리고 바르케라 연구원님께서 이렇게 멋진 주제 발표를 해주셨는데요. 바로 질문이